Greetings, my name is Mark Vincent and I'm a consultant medical oncologist at the London Regional Cancer Programme and Professor of Oncology at the University of Western Ontario in London, Ontario, Canada. I'm going to talk briefly about a publication which has recently appeared in Bioessays entitled Cancer Towards a General Theory of the Target. There have been other general theories before, of course, such as Keynes's The General Theory of Employment, Interest and Money and Einstein's Theory of General Relativity. Now, I don't expect my paper to have quite the same resonance as those two, but I do hope to achieve the same sort of goal, which is to bring some order in the form of a simplifying and parsimonious hypothesis into what appears to be a rather cluttered landscape, namely the sum total of all the various ways that cancer can be treated today. This enterprise was primarily motivated by an interest in what constituted rational therapy. We're constantly being told that we need to treat cancer rationally, by which is generally meant that we need to better understand the causative molecular forces constantly driving the cancer and to use drugs capable of blocking them. By implication, any other form of treatment is empirical and probably inferior. This did not seem to me to be correct, so I set about trying to think about cancer targets in general and how they related to rationality. The basic idea, which is a reductionist one, is that all good cancer targets and the therapies based on them can be classified into one of only two fundamental categories, or in some more sophisticated examples, into a hybrid of both of these two fundamental categories. The two fundamental types of target are firstly those based on causality, and secondly those acausal targets based on recognition. These two categories correspond, respectively, to John Locke's two kinds of essence: the real essence, which refers to the underlying explanatory causes, and the nominal essence, which are descriptive and used for purposes of recognition, as in his 1689 essay concerning human understanding. Causal-based targets, responsible for the day-to-day behavior of the cancer cell, are commonly referred to as drivers. They can be subdivided into the drivers proper, i.e. the well-known collection of oncogenes, tumor suppressor genes, and probably aneuploidy, and the enablers, such as the downstream slave systems which construct the biomass of the proliferating mass of tumor cells, and the non-oncogene dependencies, such as the remaining DNA repair systems, ROS scavengers, and the mediators of local immunosuppression, all of which rescue the cancer cell from the excesses of its egregious business model. This chain of causality can be understood as consistent with Aristotle's views on causality, especially the material cause, which is the biomass of the cancer, the formal cause, the blueprint in the altered DNA, so far generally undruggable, and the efficient cause, the slave systems and enablers downstream of the entrenched DNA abnormalities, where most of the causality inhibition opportunities arise. The acausal, or recognition-based targets, can be very simply subdivided into the crude physicochemical differences between cancer cells and normal tissues, called markers, and signatures, which are more abstract functional differences between cancer and normal tissue. All the various treatments which do not work by blocking causality, in fact work by exploiting either a marker or a signature. Now, therapies directed at either of these targets, the causal or the recognition-based, are obligated to achieve two goals: efficacy and selectivity. Efficacy means the reduction in the projected number of cancer cells, and selectivity means the avoidance of intolerable toxicity, by which is meant damage to critical normal tissues and cells. It can be seen from the 2x2 table in Table 1 that these two required goals are achieved in completely different ways by causally based and acausally based therapies. For causal targets, efficacy is achieved by inhibiting the causal chain that maintains the malignant phenotype. We shall encounter a few examples shortly. Selectivity, in this case, is meant to arise because the causal chain is relatively confined to the cancer cells. Unfortunately, some of the causal mediators of malignancy are also functional to some extent in normal tissues, so causality inhibition does not guarantee lack of toxicity. However, to the extent that cancer cells depend more on these pathways than normal cells, some degree of selectivity can be obtained. A good example is imatinib in chronic myeloid leukemia. With acausal recognition-based targets, it is easiest to start with selectivity. The selectivity arises because there is an abnormal marker or signature in the cancer cells which distinguishes them from normal cells. The efficacy occurs because some otherwise relatively non-specific destructive force, for example, chemical poisoning, radiation, physical cutting, etc., is brought to act by some proximity device to the cancer, such that the normal cells are not exposed, or if exposed, are protected in some way. Thus, selectivity and efficacy are not automatically related to one another, as they are in the previous example of causal inhibitory targets. In the case of these acausal targets, there has to be some act of human ingenuity or inventiveness to link them together. The simplest examples are physical treatments like surgery or radiotherapy, which use the tumor mass as a physical marker to selectively destroy the cancer. Chemotherapy, by the way, despite its bad press, is somewhat targeted by the inability of cancer cells to repair DNA damage as well as normal cells. Some more sophisticated treatments contain elements of both causal and acausal mechanisms. The PD-1/PD-L1 inhibitors, or checkpoint blockers, are a good example. PD-L1 is a mediator of the malignant phenotype by virtue of its downstream position in the oncogenic chain of causality. It enables the cancer cell to thrive by downregulating the immune reaction that would otherwise kill it. Therapeutic blockage of the PD-L1 or PD-1, its receptor on the lymphocyte, is therefore a type of driver inhibitory strategy. However, the immune system recognizes the cancer cell as foreign by virtue of its multiple neoantigens, most of which likely have nothing to do with driver alterations and are simply passenger alterations. Therefore, checkpoint inhibition is a hybrid causal-acausal type of therapy, and this may account for its high degree of efficacy. The publication shows us that targets may be fragmented into different functions, which do not necessarily reside physically in or on the actual cancer cell. The separate target functions include cytoreduction, modulation, and cytoprotection. Cytoreduction is biomass denial or reduction in the number of cancer cells, either by the driver inhibitor or the marker signature strategy. The physical target here is usually in the cancer cell, although in the case of hormonal or nutrient withdrawal strategies, it may be away from the cancer cell itself. Modulators are agents which, by exercising a guidance, resistance reversal, or potentiation role, improve the therapeutic index of other treatments. Bevacizumab is an example, and its physical target is actually secreted by the cancer cell into the extracellular milieu and bloodstream. Cytoprotective agents act on normal tissues as a shield or compensatory mechanism in the face of otherwise non-specific toxins, for example, fulvestrant protecting the myeloid series from chemotherapy, or amifostine protecting normal tissues from radiation damage. A key point here is that modern cancer therapy is often a weapons system with multiple cytoreductants, modulators, and cytoprotectants, and their actual physical targets both on and off the actual cancer cells. The case in point is the modern treatment of advanced non-small cell lung cancer. One of the themes of this paper is that rational treatment is not just based on causality inhibition. This is too narrow. It is rational to reduce the tumor bulk by any method that works, provided the toxicity is within acceptable limits. Rationality also demands that we know something of the mechanism of action, whether it be achieved by causality inhibition or by recognition-based strategies, which exploit specific markers or signatures. Empirical treatments, which work by unknown mechanisms, might also be perfectly satisfactory, but are not rational in the strict sense of the word. There is good reason to believe that narrow, causality-based treatments are not effective for very long, with rare exceptions, despite their intellectual appeal. Successful curative treatment of cancer will likely require overwhelming destructive force, but confined to the cancer cells. Chemotherapy, radiotherapy, and surgery have already achieved a great deal, curing many patients, and newer immunological therapies may achieve more. Causality-based treatments may assist in this exercise, but by themselves may now be experiencing the law of diminishing returns. Finally, it must be asked why this paper is entitled Towards a General Theory. Until we can know, in advance, for each cancer in each patient, what steps are required to cure that patient, this theory must remain incomplete. It may be that it will always be impossible to know in advance whether any body or any particular cancer can certainly be cured. For instance, consider a small, solid tumor that is surgically removable. We can only know this will be curative for sure if it can also be known that no viable micrometastases exist, and this will always likely be impossible. It can never be proven that the needle is not in the haystack. Likewise, even if we know the molecular composition of all the cancer cells included in a biopsy specimen and could design surefire targeted therapies, we would nonetheless never know the molecular composition of all the other cells which may or may not still be in the patient's body, given the high rate of stochastic mutations. Therefore, this approach might also be theoretically impossible. The best we can hope for is empirical probability estimates of cure rates based on large data sets, but this is not a theory. Therefore, it may be that a general theory of the target will always be incomplete. Thank you for listening, and I hope you find the paper stimulating.